Welcome back, everybody. My name is Andrea Wilson from the Roslin Institute, and it's my pleasure to introduce the next three speakers for the next session. Our first speaker is Claire Guinat. Claire is a Marie Curie Research Fellow at the ETH Zurich in Switzerland. And today she will be talking about disentangling the role of poultry and wild birds in the spread of high-pass avian influenza in Europe. The floor is yours.
Okay, can you hear me?
Yes.
Yeah, and you can see my slides?
All is good, yes.
Oh, okay, all is good, okay. So, hi everyone. So I'm going now to present you the study on which I'm currently working and which aims at disentangling the role of poultry and wild birds in the spread of highly pathogenic avian influenza virus, H5N8, in Europe. So in 2016-2017, the virus caused one of the most severe and unprecedented epidemics in terms of number of poultry outbreaks, wild bird cases, and affected countries. As you can see on these figures, which shows you poultry outbreaks in blue, bird cases in orange. So most of poultry outbreaks occur in France, but also in Poland, Hungary, Czech Republic, and Germany. So several research questions remain poorly understood, and while epidemiological and phylogenetic studies have been employed extensively during the epidemic, the genetic data has not yet been used to its full potential. So using a phylodynamic framework, this study aims at addressing the following questions. So first, how fast did we respond to the epidemic? Then how many poultry farm outbreaks and wild bird cases did we miss? How fast did the virus spread among poultry farms and wild birds? So recently, phylodynamics have become a very powerful tool to quantify the disease transmission dynamics during epidemics from genetic data and epidemiological data, and have been widely and successfully applied to diverse global public health, such as COVID-19, avian influenza, rabies, or HIV infection as examples. So to do so, which phylodynamic tools do we need? So actually, obviously, we need genetic sequences from the field, then a nucleotide substitution model that describes the nature of nucleotide substitutions, then a molecular clock model that describes the rate at which nucleotide substitutions occur over time. And finally, probably one of the most important things here, and we'll mainly focus on this tool in the next slides, a population dynamic model that describes the history of pathogen population dynamics. So let's start with the genetic sequences. So in this study, all H5N8 genome sequences of HA segments were collected during winter 2016-2017 from 4 European countries, Czech Republic, Germany, Hungary, and Poland, were downloaded from this setup. And as the aim was to infer the transmission dynamics of H5N8 among poultry farms and wild birds, the original dataset was subsampled so that only one sequence was randomly selected per poultry farm and per wild bird. Selected sequences were then annotated with available sampling dates, locations, and hosts, aligned using MAFT and manually edited using Alevio. And the final dataset consisted of 190 genome sequences from infected poultry farms and 130 from infected wild birds. So this graph shows you the temporal distribution of the number of sequences in blue for poultry farms and orange for wild birds that were collected over the number of poultry outbreaks and wild bird cases that are illustrated here in gray and reported to MPRESI. So I will skip the second and third tool and move directly to our tool of interest, which is the population dynamic model here. So the multi-type birth death model was fitted to the sequence alignments. So what does that mean? So this model especially allows for the direct estimations of key epidemiological parameters so that the transmission death rates and provide RE, so the effective reproduction number estimates that are comparable to those inferred by mathematical models. They also allow explicit modeling of the sampling process through a sampling proportion parameter. And finally, they allow quantifying transmission rates in structured populations. So under this model, sequences were organized into 5 discrete subpopulations, so according to the host type and the geographical location. So poultry farm in Czech Republic, poultry farm in Germany, poultry farms in Hungary, poultry farms in Poland, and wild birds in the 4 countries. So all sequences from wild birds were aggregated into one subpopulation and not a country-related subpopulation as for poultry, since we assumed that wild birds could move freely among countries. So under this model, infected hosts could transmit the virus to another host from the CEP subpopulation with the rate lambda, eventually become non-infectious due to recovery or death with the rate delta, be sequenced and sampled upon becoming non-infectious with a proportion S, and thus are included into the dataset we have. Or also, infected hosts could also transmit the virus to another host from another subpopulation with the rate lambda. So a few comments on the model priors and assumptions. So it was assumed that once sampled, a given host could not be infected and sampled again, since infected poultry farms were subject to culling following the confirmation of infection. And sampling of wild birds was from a mortality event. Then the origin of the tree was associated to subpopulation of wild birds in the 4 countries, since the emergence of H5N8 virus in Europe was likely attributed to infected migratory wild birds coming from Northern Eurasia, which leads to occasional or multiple viral incursions into poultry farms. For each subpopulation, the sampling proportion prior was set to capture the fact that some poultry farm outbreaks and wild bird cases could have been missed during the epidemic. The RE was estimated across 4 time intervals corresponding to 4 phases of the epidemic. And finally, all transmission become non-infectious and sampling processes are assumed to be subpopulation specific and constant through time. So the phylogenetic analysis was implemented in the BDMM prime package using BIS2 and the Beagle library to improve computational performance. All analyses were run for 100 million steps using 1 Markov chain, and states were sampled every 10,000 steps. So the first 10% of steps were discarded as burn-in, and convergence was assessed in tracer by ensuring that the estimated sampling size values associated with the estimated parameters were all above 200. Accordingly, we can obtain phylogenetic trees that are structured, which means they are associated with a specific subpopulation along their branches. And what we can infer from these structured trees are the epidemic trajectories. So to help you understanding what are these trajectories, I adapted a figure from Tim Vaughan et al., where we can see at the top the full transmission tree, which contains information on when infection happens and between which lineages, and when individuals were removed. So the sampled transmission tree in red represents a subset, only a subset of the full tree, while the rest of the transmission tree in blue is unobserved. So the epidemic trajectories correspond to the sequence of transmission, become non-infectious and sampling events that occur during a given epidemic. And actually, as you can see on the graph at the bottom, there is a group of trajectories in this example, for example, E1, E2, and E3, that are compatible with a sampled transmission tree. The structure trees were inferred by applying a stochastic mapping algorithm implemented into the BDMM prime package to a subsampled set of trees and posterior model parameters. Then the maximal credibility tree was obtained from the structured tree in tree annotator and annotated using the GG tree package in R. And finally, epidemic trajectories were simulated based on an adaptive toe-leaping algorithm. So once we have the epidemic trajectories inferred from the sequence data, we can select the events of interest in each trajectory and summarize its time and location. So in this histogram, we selected the first imported outbreak for each trajectory that represent a transmission event from wild birds to another poultry farm subpopulation and plot them according to the dates. So while the first reported poultry farm outbreaks was on the 1st November 2016 in Hungary, here represented by the red dashed line, the epidemic may have started earlier but been undetected. The median date of the first poultry farm outbreak in the 4 European countries was inferred on the 5th of September 2016, so showing a delay of approximately 2 months between the first inferred introduction and official case report. So more specifically, this figure shows you the temporal distribution of the inferred date of the first key epidemic events per subpopulation. So there are 3 types of events. So the first importation from wild birds, the first local transmission within each subpopulation. So which means if we are in poultry farm in Czech Republic, it means farm to farm transmission. While for wild bird, it means wild bird to wild bird transmission. Then the first exportation from each subpopulation. So one poultry farm, let's say in Czech Republic, infected another farm in another country, or a wild bird more generally. and the first no longer infectious outbreak or case. So again, here, what you can see on this graph is that the time estimates of the first imported outbreak was earlier than the date of the officially reported outbreak, depicted here as a red dashed line, so in each subpopulation. And if you look at this light green area, overall, all events show the delay, which was longer for Czech Republic as compared to other countries. And this being potentially explained by the fact that outbreaks in Czech Republic mainly affected backyard farms, while in Germany, Hungary, and Poland, it mainly affected large-sized poultry farms. Moreover, the first outbreaks arising from local transmission and exportation started before the first outbreak was officially reported in each poultry farm subpopulation, and seem to all occur very shortly after the first importation. So from the epidemic trajectories, a direct estimate of the total number of outbreaks and cases can be extracted and summarized in time per subpopulation. So here we use the cumulative number of no longer infectious outbreaks and cases in log scale from the trajectories as the most representative model estimate for the total number of infection. And we compare these numbers to the one obtained from InPrecise, which is here again plotted as a red dashed line. So we obtain a wide range of possible total number of infection in the analyzed trajectories, as you can see represented by the credible intervals. But overall, all credible intervals contain the values from the officially reported outbreaks and cases, except at the beginning of the epidemic in Czech Republic again, where the difference between reported outbreaks and our estimates shows a delay of several months. So this figure illustrates the posterior distribution for the RE associated within each subpopulation, together with the prior for comparison. And not that these are the farm-to-farm and wild-bird-to-wild-bird RE estimated across 4 time periods, namely October to November 2016, December 2016, January 2017, and February to May 2017. And the median posteriors for the majority of subpopulations here lie above 1 in at least one of the 2 first time periods, while a general decrease was often observed in some period of time depending on the subpopulation, and which likely illustrates the implementation of effective control strategies for poultry outbreaks. So according to vet authorities, the increase in RE in the 4th period in Germany could be explained by the onset of a large outbreak they had in turkey holdings, which caused approximately 25% of the total number of outbreaks. And also in Hungary, the median of 1.4 for the first period was likely related to the time when most of the outbreaks occurred in this country. So to wrap up on what we found in this study, so phylogenetic tools can be used to provide very exhaustive results to describe an epidemic, but can be computationally expensive in our case. We showed earlier outbreak origins and unofficially reported outbreak and case data with higher delay for Czech Republic. We also inferred value for the total number of infections that were relatively close to those officially reported with higher delay for Czech Republic. And finally, the RE estimates can be used to inform predictive models of disease transmission. So when appropriate pathogen genetic and epidemiological data is collected, we think that phylogenomics can inform disease transmission dynamics within and between different subpopulations with quite high level of accuracy. However, it's possible in our case that the wild bird subpopulation was structured since it combines different types of species that can be sedentary or migratory, as well as can behave differently following infection. However, more information would be needed to allow more structuration. Moreover, more epidemiological information is also needed to understand why we get such a long delay in case reporting for Czech Republic. So actually, sensitivity analysis is in progress with only a subset of sequences and different priors. And actually, future steps, just to inform you about the future steps of this study, we are about investigating which types of transmission events gave rise to poultry farm outbreaks. And with that, I would like to thank all the colleagues who have been, who are associated in this work, and I'm happy to take any questions you may have. Thanks.
Many thanks. That was a great talk. So there's already some questions in the chat. I also have some questions, but I start with the questions in the chat. I mean, first the observation, it's quite scary what phylodynamics nowadays can do to governments and to see what they fail to do. So Mart de Jong's question was, what can we learn from this to fight these outbreaks?
I think it's so it's a very, very good question, how we can use this tool to have something more applied in the field. And actually, I think the last question we are actually trying to answer, the one that I was explaining in my summary, exactly on how about investigating which type of transmission gave rise to poultry farm outbreaks, for example, because in that case, depending if you have, if we find, for example, that local transmission was had a higher contribution compared to importations. So this will have like high consequences about the strategies you will have to implement, which means local, if it's more related to local transmission, for example, you will emphasize, you will reinforce, for example, biosecurity between farms. While if you notice that your phylodynamic methods show you that most of the poultry farm outbreaks were due to importations, which means you will have to put strategies at the border. Yeah, so this is how this can have impact on the policy making and things like that, yeah, I guess.
Interesting. Mark, do you want to follow up on that? Sorry, I forgot.
Yeah, so no, I was just thinking that it's very important to have this discussion. And as we see every year introductions, I mean, we see every year the virus in wildlife, and we see introductions into poultry farms. And in the Netherlands, we have made lots of biosecurity measures, but even yeah, we still see introductions in. So we really puzzled what to do to prevent this happening. I mean, it's really not acceptable, I think, in the long run, to have every year to kill farms because you have introductions. So I was just trying, I mean, it was a great talk, but I was just trying to say we should also have this discussion with people more to the application to see what we can do. And so one of the things that of course we are thinking of is vaccination of all poultry. And so we could think about whether that is needed or whether we see other options to mitigate this situation.
Yeah, I see.
I think that would be great to pick up on the plenary discussion session, because these are really important questions. So yeah, maybe we can come back to that. The next two questions, I think I want to start with my question because they're all related. So you mentioned at the beginning that you took one sequence per farm, and then so the question there is, would it be better to have more sequences per farm, or is it just then, do you just introduce a sampling bias? And then Luigi's questions follows on from that. Did you use the entire genome sequence? And so how did you consider the reassortant strains? So yeah, how does the sequence information you have play with your results?
So actually, regarding the first question, so we selected one sequence per poultry farm because the aim was to infer transmission parameters that explain farm-to-farm transmission. So actually, we completely discarded the within host diversity, which can be present like within a farm. You can have different sequences, obviously. And so it could be possible to integrate several sequences per farm. In that case, that would mean that would be able to infer like bird-to-bird transmission rate within a farm, and also like, and also like a transmission between farms. But this is not something we have implemented yet. So we have decided to just focus on the farm level. And if we can include more within farm diversity, if it is possible, we'll probably do in another step here. And then regarding the reassortment, so actually because we had a lot of reassortment of genomes between these different countries, actually, we decided this prevented us to use several segments, and that's why we focused the study on the HA segment. So we were forced by the fact that there is reassortment that is not yet possible to be included in this analysis. However, there are like a new, I know that there are people that are trying to integrate this reassortment to improve the tree genetic process, but this is not something we are currently doing yet here. So yeah, we just took like one segment.
Okay. Pauline had a question. Pauline, do you want to ask it directly?
Yeah, it was really interesting, and it's really a naive question, but I was wondering why you divided the birds per country for the domestic flux, but not for the wild birds. Is there any reason for that? Because I guess you have the location. So
yeah, we have the location of sampling, but the fact is we're a bit confused about the fact that wild birds can actually move freely between different countries. So actually, in our sequences from wild birds, the majority is mallard and swans, and we know that both of them can be either sedentary or migratory. So we were a bit uncomfortable to say, let's say this swan or this bird that has been sampled in Czech Republic belongs to Czech Republic, while maybe it was at the border of another country. And so that's why we decided to combine all of them. But one step we'll do, like in the final steps of the analysis to check that everything is working, we'll probably try by trying this additional structuration, but I'm a bit scared that adding more subpopulations will slow definitely the inferences.
Yeah.
And actually in the MCC tree, so the summary tree we get from this analysis, we can see that some sequences from birds are embedded into different sequences from poultry farms in the same countries, which could suggest that some birds actually belong to a country. But as it is a summary tree, we are not really confident about this assumption. Yeah.
Okay, great. We have time for one more question, and that's an interesting one from Thomas. Do you want to ask this yourself, Thomas Rosendahl?
Sure. For some reason, my video isn't working, but you hear me, I guess.
Yeah, we can.
Yes.
I was just wondering about if you could comment on the potential of this use of this method for measuring the relative importance of several zoonotic reservoirs of disease. Like if you imagine that you have salmonella from that could come from pigs, or it can come from cattle, or it could come from poultry, and humans get sick from this, and you sort of have these subpopulations, and we know that we often have data about sequencing, and we have some limited understanding of spread, or we could estimate spread parameters and then use that to do a so-called source attribution. Has that ever been considered?
Yeah, exactly. Yeah. So this is one of the amazing things regarding this method, I guess, is really about the structuration of your sequences. So basically using this multi-type burst test model, you can structure your population. So it means in your case, you could actually say, I have this group of sequences coming from pigs, from humans, for example, and then you could run the analysis to infer the origin of the transmission between your different sequences. And actually one major point is really about the structuration, which allows you to estimate parameters within each subpopulation, which means you would be able to know how much this pathogens transmit between pigs, between humans, and also from pig to human and from human to pigs.
Oh, exciting.
Okay. I mean, obviously it's not as easy as I'm saying now, but I guess there are some promising studies that can be done.
Okay, great. Many thanks again for a really fascinating talk.